Welcome to BioBits and Bytes. Have you ever wondered if we could literally print a new organ, much like we print a document, or perhaps grow a tiny functional mini brain in a dish? It sounds like science fiction, right?
It really does.
But the truth is, biology and engineering are colliding in ways that are, well, radically transforming medicine.
Absolutely. It's a fascinating intersection.
Today, we're diving deep into some of the most cutting-edge fields: 3D bioprinting, organoids, decellularized tissues, and induced pluripotent stem cells. Big topic.
Huge topics. And they all tie together in really interesting ways.
So our mission for this deep dive is to unpack these complex biological engineering concepts, reveal their incredible potential, and discuss the real-world applications, and of course, the challenges that still lie ahead.
Yeah, the hurdles are significant too.
By the end, you'll be well informed on this revolutionary science. We've pulled from diverse sources, everything from YouTube lectures on tissue engineering to really in-depth academic reviews on transplantation and regenerative medicine.
A good mix. So ready to get started.
Let's start building. It's truly a moment where we're moving from simply observing life to actively designing and constructing with its fundamental blueprints.
It's a profound shift, isn't it? A totally different way of thinking about biology.
Building with blueprints. I love that analogy. So where do we even begin with this idea of building living tissue? Let's kick things off with 3D bioprinting.
Okay.
Most of us are familiar with conventional 3D printers, the ones that create plastic objects. How does bioprinting differ when you're working with something as fragile and complex as living cells?
Right. That's the core distinction. While both are additive manufacturing processes building layer by layer, essentially, conventional 3D printing uses inert materials, creates cell-free structures. Think of it like making a simple plastic mold or a shape.
Okay, like a basic scaffold.
Exactly.
Yeah.
But 3D bioprinting, well, that's about combining living cells directly with biomaterials. Sometimes you're even blending in essential supporting components like growth factors.
Growth factors.
Like nutrients. Sort of, yeah. Things that help the cells grow and organize. The goal isn't just a structure, it's a living, functional tissue.
So the ink itself is alive, or at least contains living components. That brings us to bioinks. What exactly are they, and what makes a good bioink? It sounds incredibly challenging to get that right.
You've hit on the central challenge there. The bioink is the material that carries and nurtures the cells as they're deposited, layer by layer. Ideally, a bioink needs to be a bit of a biological chameleon. It shouldn't require harsh pre- or post-processing like chemical cross-linking,
Right.
stuff that could harm the cells after printing.
Right, you need to keep the cells happy.
Precisely. It needs to hold its shape immediately after it's printed, too, to maintain the desired structure.
Yeah.
And crucially, the cells need to be distributed perfectly homogeneously within it, evenly spread,
And without being damaged in the process.
without being damaged. That's key.
That's a lot to balance.
Yeah.
Embedding living cells, keeping them viable, preventing bubbles. It's like trying to bake a cake with microscopic living ingredients that are also, well, very delicate.
That's a good way to put it.
Let's pause and simplify that part again. How do they manage to get the cells distributed evenly and precisely without essentially squishing them or damaging them as the material flows through the printer?
Okay. That's where the science of flow, what we call rheological properties, becomes absolutely critical. Cells are remarkably sensitive to shear forces. Imagine them being stretched or squished as they're pushed through tiny channels or nozzles.
Like pushing them through a very thin straw.
Exactly. So researchers must design bioinks and printing processes that keep those shear stresses incredibly low. And yes, bubbles. Bubbles are a nightmare.
Where bubbles?
They disrupt the flow, leading to uneven prints, gaps, and potentially dead spots in your developing tissue where cells didn't get deposited or can't get nutrients.
Okay, so it's not just about the biology of the cells, it's also about the physics of the fluid. Can you break down those key rheological properties for us, maybe like your ketchup analogy?
Absolutely. Think of it like this. Viscosity is how thick or runny the ink is. Too thick, it won't print. Too runny, it won't hold its shape.
Yeah.
Simple enough, right?
Got it. Thickness.
Then there's shear thinning. This is the ketchup property. It's thick in the bottle, but when you shake it or squeeze it, apply force, it suddenly becomes more liquid.
Ah.
For bioinks, this means the viscosity reduces when it's forced through the nozzle.
Why is shear thinning desired for 3D bioprinting?
Because the fluid is forced through those tiny nozzles.
Yeah.
Right. So reduced viscosity at that point prevents clogging and, crucially, reduces the damaging forces on the cells.
Makes sense. Less stress on the cells.
Exactly. Then you have yield stress. That's the minimum force you need to apply to get the ink to start flowing at all, like the initial push to get toothpaste out of the tube.
Okay.
The shear elastic modulus determines how well the hydrogel deforms under pressure and then recovers its shape. Does it bounce back or just smear?
Right. Structural integrity.
And finally, surface tension influences how the material forms neat droplets or a smooth, continuous line when it comes out. No messy splatters. All these properties need to be perfectly balanced. It's like tuning an instrument so the biobatter prints a living structure correctly without clogging the nozzle or damaging the precious cells.
That ketchup analogy really clicked for me. So if shear thinning is so important, what happens if a bioink doesn't have that property, or if these other properties aren't just right? What are the immediate consequences?
Well, you'll immediately run into problems, big ones. The nozzle will clog constantly, you'll get inconsistent droplet sizes, or the printed structure will simply collapse on itself like a wet sandcastle.
Mess.
A total mess.
Yeah.
And worse, the cells themselves could be damaged or even killed by excessive forces. That means low cell viability in your final printed tissue, which of course means it won't function. It's a delicate dance to get that Goldilocks zone just right.
Understood. So we've got the perfect bioink, hypothetically. Now, what about the printers themselves? What kind of scale are they printing at these days? Are we talking something visible or much tinier, like cell by cell?
The precision is really astonishing now. We're talking resolutions down to a few micrometers. That's smaller than the width of a human hair.
Wow.
And yes, with specialized techniques, particularly using laser technology, it's absolutely possible to place single cells, each droplet containing just one cell.
Single cells, that's a level of precision that just blows my mind.
It allows for incredibly intricate and high-density cellular structures, building tissues with remarkable detail.
Are there any particular printing techniques that stand out for this kind of ultra-precise work?
Yeah, beyond the more common extrusion-based methods, which are like squeezing toothpaste, one of the most promising techniques is laser-induced forward transfer, LIFT.
Lift here. Okay, what's special about that?
What makes LIFT so interesting is that it's a nozzle-free approach.
No nozzle. How does that work?
Basically, a laser pulse creates a vapor bubble that pushes a tiny droplet of bioink with cells onto the receiving surface. This completely bypasses the clogging problem you often get with very viscous materials or high cell densities.
Cover.
It's fantastic for achieving super high resolution, literally single cells per droplet, potentially, and you can build incredibly dense cell structures with it.
But what are the downsides? Sounds almost too good to be true, especially for those tricky materials.
Good point. There are always trade-offs in engineering, right? The primary disadvantage of LIFT is the risk of what's called photonic cell damage. The laser itself can actually harm the cells if the energy isn't precisely controlled.
Okay, so the tool can damage the material.
Exactly. It's also not easily scalable for printing larger tissues or organs just yet, and the equipment can be quite expensive, which makes it less accessible for, say, large-scale production right now.
So despite the challenges, we're already seeing some real applications, right? You mentioned tissues already printed. Grafts like tracheal splints, heart tissues, cartilage.
Yes, those are definite breakthroughs. Creating these custom grafts is a major step, and perhaps even more importantly right now is the development of complex tissue models for research.
Models for research.
Yeah, for drug discovery, toxicology testing, things like that. These bioprinted models provide a much more accurate representation of human biology than, say, traditional 2D cell cultures growing flat in a dish.
So better testing before going to humans or even animals.
Exactly. It means we can accelerate our understanding of diseases and how drugs respond in a far more relevant context, potentially reducing reliance on animal models, too. It's a huge step towards more effective and personalized medicine.
What do you think about the potential of these initial applications? It feels like we're on the cusp of something truly monumental, doesn't it?
It absolutely is. It's not just about making spare parts. It's fundamentally changing how we do research and develop treatments, making the whole process better, faster, more accurate.
Okay, so beyond printing cells layer by layer, I know scientists are also leveraging existing natural structures. You called them nature's blueprints. Let's talk about decellularized scaffolds, these top-down approaches. What's the fundamental concept here?
Right, the top-down approach, it's incredibly clever, actually. It involves taking existing tissues or even whole organs, maybe from animals, maybe human cadavers, and through a careful process called decellularization, removing all the original cells, everything living.
So you wash away the cells, what's left?
What's left behind is just the extracellular matrix, or ECM.
And if I understand correctly, the ECM is essentially the natural scaffolding, the sort of structural network that cells normally live on and interact with.
Precisely. It's the intricate, supportive network outside of the cells themselves, proteins, collagen, things like that. What's fascinating here is how this decellularization method often preserves the complex vascular networks, the blood vessel plumbing, basically.
Ah, the plumbing. And why is preserving that original intact plumbing system so crucial for regenerating organs or complex tissues? It's absolutely paramount. Blood supply is the lifeline for any living tissue, right?
Ah,
When you recellularize these scaffolds, meaning you add new cells back onto them, having that intact pre-existing vascular network allows for efficient perfusion. Oxygen and nutrients can get delivered deep into the tissue, and waste products can be removed.
so the new cells can actually survive and thrive.
Exactly. Without it, any thick tissue beyond just a few cell layers would simply die from lack of supply. We've seen incredible examples already, like using decellularized heart ECM, maybe processed into a hydrogel, to improve heart function and reduce scarring in animal models of ischemic heart disease.
Wow.
And even non-cardiac ECM, like human amniotic membrane, has been used as patches for damaged hearts, showing it can promote new blood vessel growth angiogenesis and reduce scar tissue formation.
So it's like using nature's own intricate plumbing system, then repopulating the house with new cells. What are some of the major challenges in this decellularization process? It sounds like you're trying to take apart a very delicate structure without destroying it. You're right, it's a real tightrope walk. There are several challenges. It's difficult to perfectly preserve the delicate microarchitecture while effectively removing all the original cells and, importantly, all their DNA. Any leftover DNA can cause immune reactions.
Okay, immune rejection is still a risk.
Yes, and you also need to minimize other immunogenic factors, things like specific proteins from animal tissues like alpha-gal or maybe human leukocyte antigens from donor human tissues. These can trigger immune rejection if that scaffold is eventually transplanted.
Alpha-gal, that's the one involved in that tick bite allergy, right?
That's the one. Comes from non-primate mammals. And sometimes the harsh chemical or enzymatic treatments used in decellularization can inadvertently cause structural changes in the microvasculature, which can then hinder that crucial recellularization step later on.
So it's a delicate balance. Get the cells out, but don't wreck the house.
Precisely.
Speaking of surprising sources for scaffolds, I have to ask this. Have you ever wondered why researchers are looking at spinach leaves for organ repair? It sounds like something straight out of a kid's science project.
It does sound a bit out there, doesn't it? But it's a very real and potentially a very promising area. Plant-based scaffolds. Researchers are actually decellularizing plants, things like apple tissue or, yes, spinach leaves. They wash away the plant cells.
Leaving behind the plant structure.
Exactly, leaving the cellulose fiber scaffolds. And these plants inherently possess porous and vascular structures, the veins in a leaf. They remarkably resemble mammalian vasculature.
You're kidding. Spinach mains like blood vessels.
The structure is surprisingly similar in terms of branching and scale. It makes them excellent, readily available candidates for tissue engineering scaffolds.
That's astonishing. So what are the big advantages of using plants over, say, animal tissues for these scaffolds, beyond the obvious, wow, it's a spinach leaf factor?
The advantages are pretty significant. They seem to offer good biocompatibility, meaning human cells can potentially grow on them okay. Their production cost is incredibly low compared to animal sources.
Cheap and readily available.
Right. And they're free from many of the ethical concerns and supply constraints associated with using animal tissues. The decellularization process for plants is relatively simple too, which might make them suitable for large-scale production eventually. They hold great prospects for regenerating tissues like heart, bone, maybe cartilage. It's a fascinating, sustainable pathway.
That's a huge step towards accessibility, isn't it? Low cost, easy to get. But I imagine there are challenges with using plant material, too. Our bodies aren't exactly designed to integrate spinach, structurally speaking.
Absolutely. You're right to be skeptical. Immunogenicity is still a concern. Leftover plant proteins could potentially trigger immune responses in animal systems, and macrophage infiltration, a sign of immune activity, has been observed in some studies.
So the body might still attack it.
It might. Biodegradability is another issue. Cellulose, which makes up most of the plant scaffold, is quite hard for animal enzymes to break down in vivo. Our bodies just aren't equipped for it.
So it might just sit there.
It might persist for a very long time, which might not be ideal depending on the application. Also, the decellularization process itself can sometimes damage the mechanical properties of a plant scaffold, making it weaker. And residual detergents used in the process, like SDS, can be toxic to cells if not thoroughly rinsed away.
Okay, so promising, but still hurdles to overcome.
Got it.
So we've talked about the ink, the cells and biomaterials and the scaffolds, both natural and plant-based. But what does all of this mean for getting enough cells to actually build these tissues, whether we're printing them from scratch or recellularizing a scaffold?
Ah, yes, the cell source problem.
We need a source of cells that can grow, multiply, and crucially differentiate into whatever specific cell type we need, right? Heart cells, liver cells, neurons.
That's where induced pluripotent stem cells, or iPSCs, come in. They are true cellular superheroes in this story. This was a massive breakthrough, really Nobel Prize-winning work, pioneered by Shinya Yamanaka back in 2006.
Right, Yamanaka factors.
Exactly. Essentially, iPSCs are adult somatic cells, like readily available skin cells or blood cells that have been reprogrammed. They're pushed back genetically to an embryonic-like pluripotent state,
Pluripotent meaning.
meaning they have the incredible ability to differentiate into virtually any cell type in the body, from a heart muscle cell to a neuron to a liver cell. Almost unlimited potential.
That's incredible. So we can essentially take a patient's own skin cell, reprogram it, and then turn it into, say, heart cells to repair their damaged heart. That sounds like it would completely solve the problem of immune rejection in transplants.
Exactly. That's one of their biggest, most revolutionary advantages.
Yeah.
Because they're patient-derived or autologous, the body recognizes the resulting cells or tissues as self.
No foreign tissue.
Correct. This drastically minimizes the risk of immune rejection when these cells are used in therapies. That's a major, major hurdle in current organ transplantation that requires lifelong immunosuppression.
Which has its own nasty side effects.
Absolutely. Plus, with iPSCs, you can start with easily accessible cell sources, a small skin biopsy, or blood draw, and they have the potential to generate vast quantities of various cell types needed for large-scale tissue engineering. It addresses both the rejection problem and the supply problem.
That's a great question. Let's break it down. How exactly do they reprogram these adult cells back to that embryonic-like state? Is it incredibly complicated, or is it getting more streamlined now?
It is complex, but the methods are continually improving and getting more efficient and safer. Initially, reprogramming involved using viruses. Specific viruses were engineered to deliver the key reprogramming factors, those Yamanaka factors, essentially genetic instructions, into the adult cells.
Viruses sound a bit risky for therapy, though.
Well, that was the concern. While effective, these viral methods often integrate into the host cell's own DNA. That raises safety concerns, like the potential risk of activating cancer genes, causing tumor formation, or leading to unstable gene expression down the line.
Okay, not ideal for putting back into a patient.
Definitely not ideal. So, to address this, non-integrating methods have emerged and are now preferred for clinical translation. Things like using episomal plasmids, small circles of DNA that don't insert into the genome, or even delivering the reprogramming factors as mRNA molecules directly.
mRNA, like in the vaccines.
Exactly like that concept. These are considered footprint-free because they don't permanently alter the cell's genome. They do the reprogramming job and then disappear, making them generally safer for potential clinical applications. It's always about finding that balance between efficiency and safety.
Makes sense. Safer is better when it comes to therapy. Now, let's shift gears slightly and talk about organoids. These mini organs seem to be popping up everywhere in research news lately. They sound amazing, truly blurring the lines between lab bench and living biology. What exactly are they, and how do they even make them?
Organoids are just fascinating. They're 3D structures grown in vitro in a dish or bioreactor, usually starting from stem cells, including those iPSCs we just discussed. The amazing part is that these cells self-organize. They arrange themselves and differentiate to mimic the complex architecture and often, to some extent, the function of actual human organs.
So they build themselves, almost.
In a way, yes. You provide the right starting cells in the right environment, the right soup of growth factors and matrix, and they follow their inherent developmental programs to form these organ-like structures. Think of them as tiny, simplified versions of a liver or a brain or a kidney or intestine grown in the lab.
Wow, mini organs.
They represent a significant leap beyond traditional 2D cell cultures, which are just flat layers of cells lacking the crucial 3D interactions of real tissue. And they often outperform many animal models for studying human-specific biology.
If we connect this to the bigger picture, why are organoids such a game changer for medicine? What's the profound shift they enable compared to older methods?
They're a game changer because they bridge a critical gap. They offer unparalleled advantages for drug development, for understanding the mechanisms of human diseases, and even in the realm of personalized or precision medicine. For example, they are revolutionizing disease modeling. Researchers can now grow cancer organoids directly from patient tumor samples, colon cancer, breast cancer, pancreatic cancer.
Patient-specific cancer models.
Exactly. This allows researchers to study the specific biology of that patient's tumor and test different chemotherapy drugs on the organoid to see which one works best before even treating the patient. Personalized cancer therapy in a dish.
That's incredible.
It's incredibly valuable for neurological disorders, too. Think about autism spectrum disorder, multiple sclerosis, Huntington's, Parkinson's. We can generate brain organoids from patient iPSCs and try to understand what's going wrong at a cellular level in a human-relevant context, which is often difficult or impossible in mouse models.
That's amazing for understanding disease mechanisms and testing potential treatments. What about drug discovery more broadly and the regenerative medicine side of things?
For drug discovery, liver organoids are becoming really important for testing drug toxicity. They give a much better prediction of how a drug might affect a human liver compared to animal tests, potentially catching harmful side effects earlier.
Saving time and money and maybe lives.
Potentially, yes. Lung organoids, for instance, were instrumental during the COVID-19 pandemic. Researchers used them for drug screening because they accurately mimic signals from a real infected lung and helped identify potential inhibitors like imatinib much faster.
Real-world impact already.
Definitely. And in regenerative medicine, we're seeing intestinal organoids being explored to treat inflammatory bowel disease, maybe by transplanting healthy organoids, salivary gland organoids for conditions like dry mouth or xerostomia. And liver organoids are showing promise for treating certain inherited metabolic diseases, even potentially acting as a platform for ex vivo gene therapy.
Ex vivo gene therapy. How does that work?
The idea is you take patient cells, make iPSCs, grow liver organoids, correct the genetic defect in the organoids in the lab, and then transplant the corrected mini-livers back into the patient to restore function, modifying genes outside the body first.
Oh, wow. That opens up a lot of possibilities. It's a fundamental shift in how we approach research and therapy development.
It really is.
So if we combine 3D bioprinting, which gives us the structure and layer-by-layer control, with organoids, which bring that complex function and self-organization, that must be leading to even more intricate and functional structures. How does that synergy unlock new possibilities?
Oh, it's truly where the magic happens now. The synergy between 3D bioprinting and organoids is creating incredibly advanced multi-tissue models that function much more like real organs. It's the next level.
Can you give some examples?
Sure. We're seeing things like vascularized lung cancer organoids being bioprinted. These have built-in blood vessel networks, making them far better for testing drugs that need to reach the tumor through the bloodstream.
More realistic drug testing.
Much more realistic. Or bioprinted skin organoids that has integrated vascular structures, making them better models for studying wound healing or testing cosmetics. Even in neural applications, bioprinting can help guide the direction that nerve fibers or axons grow within or between organoids, moving towards building more functional neural networks, maybe for spinal cord injury research.
So it allows us to not just grow these mini organs, but to precisely place them, connect them, and build larger, more complex functional systems.
Exactly. It's about adding that next layer of organization and architecture that self-assembly alone might not achieve. Structure plus function working together.
Okay, this ties back to something we touched on earlier. A major hurdle in organ transplantation has always been immune rejection, requiring lifelong immunosuppressants, which, as we said, come with their own set of serious side effects. How do these new technologies, especially iPSCs, help address that monumental challenge?
iPSCs are really the cornerstone of addressing rejection. As we discussed, because they are patient-derived made from the patient's own cells, any tissue or organoid structure grown from them is genetically matched to the patient.
The body sees it as self.
Precisely.
Yeah.
So the immune system shouldn't attack it, or at least the risk is drastically minimized compared to receiving an organ from an unrelated donor. This holds the incredible promise of avoiding, or at least significantly reducing, the need for those lifelong immunosuppressant drugs.
Which would be a massive improvement in quality of life for transplant recipients.
Huge. Eliminating the risks associated with immunosuppression, like increased infections, certain cancers, kidney damage, would be revolutionary for transplantation.
This raises an important question, though. How close are we realistically to bypassing lifelong immunosuppressants in human transplants using these methods? Is it just around the corner, or still more of a distant dream?
That's the million-dollar question, isn't it? It's certainly not routinely available just around the corner for complex organs yet, but the progress is incredibly encouraging and rapid. While iPSCs are a massive step towards immune compatibility, researchers are also exploring even more advanced strategies to induce tolerance.
Like what?
For instance, there's fascinating work in mouse models using nanoparticles. These nanoparticles are targeted specifically to the spleen, which is a key immune organ. They can deliver signals that essentially retrain the immune system locally, making it accept the transplanted heart without needing full systemic suppression.
Wow, targeted immune training.
It's highly sophisticated immunology. This indicates that researchers are actively pursuing ways to modulate the immune system specifically towards the transplant, to induce tolerance, rather than just using blunt force whole-body suppression. The promise is significant, but there's a lot of complex biology and immunology to unravel and translate safely into humans. It's coming, but it will take time.
Okay, so progress, but patience is needed. Now, looking beyond the fascinating science and the clinical applications, what about the broader implications? Especially with organoids, growing mini brains in a dish or creating fused organoids with sensory capabilities. That can raise some pretty profound ethical questions, can't it? How do we navigate that territory?
Indeed. The ethical implications are very significant, particularly concerning brain organoids, and it's an area of very active ongoing discussion and debate within the scientific and ethical communities. The potential, however remote right now, for consciousness or some form of sentience to emerge in complex in vitro brain organoids, well, that raises deep ethical dilemmas. Questions about moral status, the potential for suffering, and where the boundaries of research should lie.
Yeah, that's heavy stuff.
It is. Similarly, creating fused organoids that might integrate sensory inputs, like responding to light, or perhaps even touch, pushes us to think carefully about responsible innovation. Organizations like the International Society for Stem Cell Research, the ISSCR, have established ethical guidelines. They emphasize careful monitoring, clear limits on complexity and duration of experiments, and adherence to responsible practices. But it's a rapidly evolving field, so the guidelines need to evolve too.
Constant vigilance and discussion needed.
Absolutely.
And what about the regulatory landscape for actually getting these groundbreaking therapies from the lab bench to patients? It must be incredibly complex to ensure safety and efficacy for something so fundamentally new and often living.
It is exceptionally complex. Cell and gene therapy products, or CGTs, which encompass many of these technologies, face unique regulatory challenges. This is because they're complex, often living products. They can persist long-term in the body, and they carry novel risks compared to traditional drugs or devices.
Who oversees this?
Agencies like the FDA in the U.S., specifically their Center for Biologics Evaluation and Research, CBER, handle the regulation. They often require extensive preclinical testing and very careful clinical trial design. And critically, they usually mandate long-term patient follow-up for these therapies, sometimes for years or even decades, because the effects can be long-lasting and potentially irreversible. We need to understand the long-term consequences.
Makes sense. But does that slow things down too much if these therapies are urgently needed?
That's the balance regulators have to strike. To help accelerate promising therapies for serious conditions, the FDA and similar agencies elsewhere do have expedited programs. Things like orphan drug designation for rare diseases, fast-track breakthrough therapy designations. These can speed up the review process for therapies that show significant potential early on. It's a constant balancing act between ensuring safety and facilitating access to potentially transformative treatments.
Caution and progress in parallel.
That's the goal.
And finally, these larger societal considerations. Thinking about genetic engineering, manipulating life at this fundamental level, these aren't entirely new conversations, are they? They have roots in historical discussions within science itself.
They absolutely do. A key example is the Asilomar Conference on recombinant DNA back in 1975.
Asilomar, right, what happened there?
That was a landmark event where scientists themselves proactively got together to assess the potential risks of the then new recombinant DNA technology, basically, gene splicing. They established initial safety guidelines for its use.
So scientists policing themselves?
To a large extent, yes. It was widely praised at the time for scientists taking responsibility. However, it also faced criticism later on. It was quite exclusive, mostly just scientists, without much broader public representation or input from ethicists or social scientists. And some argue its focus was primarily on preventing lab accidents or escaped organisms, sort of narrow scientific safety, without fully grappling with the wider ethical, social, and philosophical implications of manipulating genes right from the start.
Interesting. What's the lesson there for today's technologies?
I think it highlights the ongoing critical need for inclusivity and transparency in these discussions now. As these biological engineering technologies become even more powerful, we need conversations that involve not just scientists and regulators, but also ethicists, policymakers, patients, and the broader public. We need to ensure these powerful tools are developed responsibly, equitably, and aligned with societal values.
Open dialogue is key.
Absolutely essential.
Wow. What an incredible journey through the cutting edge of biological engineering. Just amazing stuff. To quickly recap what we've learned today, let's maybe think of it in a few key takeaways for everyone listening. First, 3D bioprinting isn't just printing plastic, it's literally building tissues with living cells as ink. And the precision, down to single cells, is just astounding.
Really mind-blowing precision.
Second, the bioinks and the printing technologies themselves are incredibly complex. It requires this delicate balance of biological needs and physical properties, but the field is advancing really rapidly.
Constant innovation there.
Third, decellularized scaffolds give us nature's own blueprints. Taking existing tissues, even surprisingly from plants like spinach, washing out the cells, and leaving that intricate structure, especially the vital vascular networks, offers biocompatible and maybe cost-effective alternatives.
Leveraging nature's designs.
Fourth, induced pluripotent stem cells, iPSCs. These are our cellular superheroes, right? Patient-specific master cells that can become almost anything, unlocking personalized medicine and holding huge promise for tackling transplant rejection.
A true game changer, the iPSCs.
And finally, organoids. These amazing self-organizing mini-organs are revolutionizing how we model diseases and test drugs. And when you combine them with bioprinting, well, that opens up even more complex possibilities for regenerative therapies down the road.
Putting it all together.
Exactly. Together, these technologies offer immense hope. Hope for solving organ shortages, for overcoming the challenge of transplant rejection, and really moving us closer to a future of truly personalized, effective medicine.
And as we look to that future, here's maybe a thought to reflect on. Consider how these BioBits and Bytes, this intersection of biology and information and engineering, might not just replace organs or fix diseases in the ways we currently think about. They might fundamentally change our understanding of what it even means to be healthy, or perhaps even what it means to be human in the coming decades. It's a profound potential shift in our relationship with our own biology.
A profound thought indeed. Definitely something to mull over. Thank you for joining again for this deep dive into the fascinating and slightly mind-bending world of biological engineering.